I'm Michaela Welch.
And I'm Hadley Eckert.
And this is our project for cloning and sequencing the gene for GAPDH. A little overview on what we did. Our objective was to PCR amplify, clone, sequence, and analyze a GAPDH gene from a plant. These were the steps that we took throughout the project in order to get the final result that we wanted. Some background on what GAPDH gene is. GAPDH is a catalyst involved in glycolysis and respiration. GAPDH protein is highly conserved between organisms, and it's not necessarily identical between every organism. There are many opportunities to draw connections between the GAPDH and its biomedical and evolutionary significance. That's why this project was super significant. So for step one was to extract the actual DNA from a plant. The plants that we chose were carrot and strawberry leaves. In order to get the cell from these leaves, we used a pestle and mortar in order to grind up the plant leaves super well. And then we used a lysis buffer, and this was used to break open the cells and disrupt the cell membranes. Then we followed that with centrifuging in order to remove cellular debris and isolate DNA in supernatant. Step two was to amplify the GAPDH gene. Amplification is creating multiple copies of the targeted segment of DNA. This way we can have a larger amount of DNA in order to follow up on with our experiment. So for step two, in order to do this, we used a PCR, which is a polymerase chain reaction. This included a master mix that we created and our template DNA. In order to find the results of our PCR, we used gel electrophoresis, and this is our round one of it. It included 4 wells that held the weight ladder, the negative control, our Arabidopsis G DNA, the PGAP plasmid DNA, our carrot DNA, and our strawberry DNA. And in this picture, you can't see it very clearly, but there wasn't any movement of our bands, which we kind of assumed with this because we didn't have a necessarily nested PCR. But we did do a PCR for nested in round 2. And in our gel electrophoresis, we were supposed to see movement in this one, but since we were unable to find any gap DNA in any of the wells, we proceeded forward with a different group's DNA. So we continued the project with a tomato plant. And for step 3 was to purify the PCR product. We used Quantum Prep PCR clean spin columns, and this is an example of what they kind of look like. This is to purify the PCR products from NTPs, primers, polymerases, and the other gunk that's kind of in there with the DNA. So we took the master mix and our template DNA and everything that was with it and put it into these little spin columns, and after a couple of centrifusions, the smaller molecules will fall to the bottom and the larger molecules will stay held in the spin column.
So our step 4 was to blend and ligate our PCR products. We went with a blend end ligation instead of a sticky end ligation because we needed less specific DNA to connect those two. And so ligation is pretty much the process of joining 2 pieces of DNA together into a single piece through the use of a DNA ligase. We use specifically the T4 DNA ligase. You can see in the image over on the right that we take a cloning vector and then we take our target DNA, which contains that GAP gene, and we basically stuck them together so that we just have one single vector containing our target DNA. And that leaves us with the cloning vector called PJET 1.2. We went on with this cloning vector and transformed our bacteria with it. The bacteria that we used was E. coli, and we used the heat shock method, which means we took our cold DNA plasmid and spread it with our bacteria cells and spread it across the hot agar plate. And we allowed that E. coli that contains our cloning vector and plasmid DNA, we allowed that to grow and replicate, so now we had more E. coli and more of our GAP gene. And you can see our E. coli agar plate there on the right, and we chose 4 random colonies to use. For our next step, which was to isolate that plasmid and analyze with a restriction digestion. And so we took those 4 bacteria colonies and we used a restriction enzyme BGL2 to produce 2 linearized fragments. And so with the gel electrophoresis, we should have seen a band with our vector DNA as well as a band with our GAPDH. Now, as you can see in our results on the right, that we did not really have any clear bands containing those. However, with our well number 2, which contained DNA from the 2nd colony, there was a very, very faint band there, and so we decided to proceed with the colony 2's DNA. We went ahead and sent that DNA to the lab in South Korea to be sequenced. So after we got the sequencing back from the lab, we used FinchDB to analyze this, and we could see our base pairs and the order that they were in. We took these base pairs and we blasted them using BLAST-N in order to find source matches to determine if that GAP gene was present in the DNA that we sent over. Our BLAST results came up showing that our DNA came from synthetic constructs or other genetic sequence, meaning that it did not contain that target GAP DNA. It basically means that you can see here our gene was the green there, and the DNA that we sent to the lab was the rest of the gene, and that was our vector. And so overall, we did not accomplish our goal of sequencing that target gene from our E. coli. Possible mistakes could have been with our original 2 plants, the carrot and the strawberry. We may not have extracted the DNA correctly in the first step with the pestle and mortar, and that's why we did not see any DNA in our first 2 PCR experiments. When we proceeded with the tomato experiment, with the tomato plant, we may not have received any DNA as well from random mistake in choosing our colonies in the bacteria. We chose those 4 colonies based off chance, and they may not have contained that replicated plasmid DNA. However, we still did learn throughout this experiment a lot of useful information about the DNA's overall structure, as well as many different laboratory techniques and methods.
Any questions?